My pleasure to introduce Sanad Divanovic, who is our first Liver Center seminar speaker of 2024. So Dr. Divanovic actually is an associate professor in the Division of Immunobiology in the Department of Pediatrics at Cincinnati Children's Medical Center, where he's actually been on faculty since 2010 or 2013, roughly. So just to give you a little bit of information about his background, he completed his undergraduate degree at DePauw University in Indiana. Then he went to get a master's degree at the University of Oklahoma, came back for his PhD to the University of Cincinnati, and then moved sort of down the road to Cincinnati Children's Medical Center for a postdoc in immunology. So it's always been his focus, immunology and immunology of the liver. Actually, in his early days, he was focused on immunobiology of preterm birth. But somewhere along the line, he shifted towards the nexus of immunology and metabolism, and that has been his focus for most of his faculty career. And he's been continuously funded by NIH in that vein since for the last 10 years. I think two of the things I'd like to mention before I give him the podium. One is that one of his seminal observations is basically the finding that IL-17 and IL-17 producing cells are present in metabolic liver disease and are integral to the pathogenesis of non-alcoholic fatty liver disease. And in addition to that, and this is one of my favorites, he made some seminal observations that housing mice at thermoneutrality is actually essential for getting them to establish some of what we consider the hallmarks of human fatty liver disease. And he's going to talk a little bit about that today. But I think, as you can see from the title of his talk, he's now turning things around a little bit from immunologic sort of drivers of dysmetabolism. It sounds like he's going to talk about dysmetabolism driving immunological alterations. And so I'm really pleased to welcome Dr. Divanovic to the podium today.
Well, thank you, Jackie, for that very kind introduction. It's a pleasure to be here, meet some old friends, and looking forward to meeting up with people and discussing science for the rest of the day. So as Jackie alluded, I'm going to give you a little bit of background on how I got into this field, why an immunologist would fit into this, and then what we're trying to understand, really what moves these immunological processes and how they contribute to the pathogenesis on non-alcoholic fatty liver disease, or MASLD as it is known now. So let's see. Let's see. Oh, there we go. We got it. Okay. All right. So the audience doesn't need much introduction, but obesity is a pandemic. The global rates of increase in obesity are expanding tremendously. And as CDC estimates, by 2030 or so, we're going to be dealing with approximately 40% of adults to be obese in the United States. So this rapid rise in obesity, obviously, it's associated with numerous diseases. The focus of today's talk will be MASLD, or formerly known as NAFLD. But my lab is also now focused on how this promotes susceptibility to infection as well, but we're not going to touch that today. Now, the reason for studying MASLD or NAFLD is that therapies targeting obesity and metabolic derangements with long-lasting efficacies remain severely limited. We know that this weight gain promotes pathological accumulation of excess adipose tissue. It leads to metabolic endotoxemia. For an immunologist, that's exciting because it obviously triggers or tickles your immune responses and probably rewires them. So understanding how that rewiring works is probably important in a disease setting. We know that this obesity is also associated with chronic low-grade inflammation, that there is occurrence of intestinal dysbiosis, and obviously, these changes have all been associated with epigenetic modifications, which is another direction of my lab, is trying to understand what are the vertically transmitted pathogenic molecules or processes that may amplify this rise in obesity. So we're going to touch upon this vertical transmission just a little bit at the end. but because we're still just scratching the surface on how to do these studies. However, what I wanted to tell you is what really my interests in this are, and that is immune repertoire and diet-induced obesity. So when I started working in this field several years ago, the key observations were that there's epidemiological studies suggesting that are associated with acute phase reactant TNF and IL-6 increases in obese individuals. Now, long time ago, Stuart Weisberg first time showed that there is increased activation and recruitment of macrophages into adipose tissue, and first seminal studies were suggesting that macrophages are the key promoters of this inflammation in obesity and drivers of the pathogenic outcomes of metabolic diseases. Now, if you go through PubMed, you can pick if you're an immunologist, you can pick your favorite immune cell, type it in there, and you're going to find that that immune cell correlates with metabolic diseases, pathogenic or not. And so essentially what you can conclude is that the obesity is associated with a broad activation of pro-inflammatory and stress response pathways, and that inflammation plays a central pathogenic role in essentially all of the adverse sequelae of obesity. So as an immunologist, this is quite exciting because we can really pick and choose what we want to study, and it's likely to have an effect. So my lab decided to focus on metabolic dysfunction-associated steatotic liver disease, or MASLD is now known. It is the most common chronic liver disease in the world. It comes in different flavors. It progresses. I don't need to tell you about this. And currently, no effective therapies exist. So as an immunologist, really the key point of my lab was to try to understand how this progression of the disease goes from a fatty liver to an inflamed liver. And premise for that is that if we can understand the immune pathways that lead this progression, maybe we can target them, because targeting immunological processes is quite effective therapeutically. So maybe we can reduce the progression, slow it down, or potentially, hopefully, even reverse it. So to do that, I don't need to tell you, but immune responses need to be tightly regulated. If you are less pro-inflammatory, you're likely to pay the consequence due to susceptibility to infections. If you're more pro-inflammatory, you may develop autoimmune disease, or you may pay the price for in context of metabolic diseases. So if you look at just MASLD right now, this is one of the recent reviews. I mean, you can pick and choose an immune cell that you want to study, and they're pretty much all associated with progression of this liver disease. So what this is telling us is we're not going to find a magic bullet. One immune cell is unlikely to contribute to all of these processes. These are highly complex details and processes that one cell communicates with another cell, and it leads to amplification of inflammatory cues. Not to mention in humans, it's likely to be even more complex because we are all genetically different. We are exposed to different environmental cues, and so these stimuli are likely to be different. But still, we can only take a reductionist approach to study something. And if you go through the literature in the last 15, 20 years, innate immune cells are or have been dominantly studied, primarily Kupffer cells or recruited monocytes and macrophages in MASLD progression. Recently, however, the adaptive immune cells have emerged as also key contributors to this. And this is really where we come in. Some people have started studying B cells as an important contributors to this. We have primarily focused on T cells and specifically on CD4 cells with primary emphasis on Th17 cells. So why are we focused on that? As Jackie alluded, one of my early discoveries was that the IL-17A, which is predominantly produced by Th17 cells, is important in regulation of MASLD progression. But there are other cytokines such as TNF, interferon gamma, IL-1 beta, and IL-6 that all have been shown to play a role. So today I'm going to try to tell you, once we establish that IL-17 works, now really the emphasis is trying to figure out what regulates this IL-17A production by these cells. And if we can hopefully figure that mechanism out, maybe there could be therapies that we can selectively target production of this in a context of MASLD without causing too much harm. So what is IL-17? Well, it's a small family of pro-inflammatory cytokines. as shown here on a side. There's only 6 or 7 family members. Was initially discovered as being a key player in a barrier immunity. Primary producers reside largely in the skin mucosal tissues and in the liver, and the receptor for this molecule is expressed by a variety of cells present in the liver. Now, human studies suggest strong correlation of IL-17 expression and progression of mast cell, and in the mouse models as well, we see that the manipulation of this axis is important in disease progression. So given the nature or knowledge that Th17 cells are important producers of this, really we wanted to see how this is occurring. So just to give you an emphasis of what we do, we study both the mast cell progression in humans and mice, but mice are really our primary focus because we can manipulate the axes to our liking. So our favorite model is really taking the black 6 mice, C57 black 6 mice, placing them on a high-fat diet, and then looking at what happens in their liver. And so as shown here, if you place the mice on a high-fat diet, as known, there's an increased accumulation over time of immune cells present or infiltrating the liver. Now, what is really nice to see is that if you take this whole liver tissue out and you do just a simple PCR, highly expressed genes that you will find in this liver tissue are genes that are important in promoting polarization of naive T cells towards Th17 cells. So that gave us a little bit of a clue that maybe Th17 cells are present in there or infiltrating into the liver. And so in fact, if you look at it in the liver on a high-fat diet, there's a lot more of IL-17 producing cells. And what we find very fascinating is that dominant producers of this IL-17 are CD4 T cells and not CD8 T cells or non-T cells. Now, this is very different in the liver compared to fat, because in a fat, the primary producers of IL-17 are gamma delta T cells. So this told us really that there's some unique components of these Th17 cells in the liver that we want to study because we would like to target this inflammation in the liver, but not necessarily in the white adipose tissue. So do T cells, the key question is, are T cells really important in mast cell progression in mice? And so to establish this study, we use the RAG2 knockout mice. These are mice that don't have endogenous T cells. And so if you place them on a high-fat diet, these mice become obese. They gain equal amount of weight as control mice. But you can see that if you do not have any in a RAG2 knockout mice, essentially, there are no T cells. And so what you can do is you can take exogenous T cells and transfer them into them and ask whether they go into the liver. And you can see that you can have a good reconstitution. If you harvest the liver on a high-fat diet of these mice, you can see that they are actually able to, these transferred T cells, they can produce, have a different variety of pro-inflammatory cytokines, including IL-17A, and that they go to the liver. And this leads to recruitment of macrophages, Kupffer cells, and neutrophils in these mice that have now received T cells. Now, what was important to this finding is that really, if you just take the RAG2 knockout mice and place them on a high-fat diet, they will develop fatty liver, but there will be no inflammation in the liver. With transfer of T cells, now you get this inflammation. It's arguing that these T cells are really important in recruitment of immune cells and progression to inflammation. And so you can see also here that if you transfer this can be evaluated physiologically by increased hepatocellular damage to ALT levels. So are T cells important in human models of NAFL or NASH or mast cell? And so the answer is yes. I wouldn't be showing you this because we have a nice cohort of adolescents who are undergoing bariatric surgery. And the beauty of this cohort is that about 25% of them don't have any metabolic disease. So they're just metabolically healthy, obese individuals. They're morbidly obese. Then we can tease out the ones that have NAFL alone, NASH, or type 2 diabetes, et cetera, different flavors. And so if you take just the perform RNA-seq on the whole liver, for example, here you can see that there's a difference in gene expression between the ones that have NAFL or fatty liver versus inflamed liver. And that predominantly this is associated with multiple pathways, but our favorite pathways, there is an increase in the liver of inflammation and IL-17 signaling pathway. So this gave us an instruction that, okay, we have some relevance in humans. Can we pursue this further? So we went on from these individuals. We also collected the immune cells from the liver biopsies that we can get, and we performed single cell RNA-seq analysis on that. And so these are metabolically healthy, or what would be equal to obese, and these are individuals with NASH. And you can see that now individuals with NASH have increased presence of CD4 T cells in the liver. And if you ask, what we found out that was quite exciting is that these T cells, although they're present in NASH individuals, they do not come in the same flavor. They are different flavors of CD4 T cells in this. And so we became quite interested in trying to figure out what these flavors are and why are they present there? Can they contribute to a disease? So to do this in a mouse model, it becomes a little bit challenging, okay? And so the reason why is because lots of these models do not fully recapitulate what you see in humans. So for example, in the basic studies with mice, there are genetic manipulations. You can do OB leptin or leptin receptor deficient mice or dietary modulations, but only certain strains of mice become obese. So C57 black 6, AKR, etc. Nude mice, even on a C57 black 6 background, meaning they have everything but fur, they do not become obese. Atherosclerosis, which is a primary mortality killer in humans with NASH, mice do not develop atherosclerosis unless you knock out the LDL or LDLR receptor for them. And obese wild-type mice are protected from hepatic fibrosis. And for what was important to us also clinically as well, looking at it, that the female mice have a hard time becoming obese unless you perform ovariectomy in this setting. So how could we change that? Can we improve these models to get us closer to a human disease? And in fact, what we found through some literature is that by changing the temperature at which we house mice could actually help us get there. All right. And so the first one is that the humans, they prefer thermoneutral conditions, meaning that you do not have to be cold or too hot, is between 20 and 23 degrees. But that's not the perfect temperature for mice. What mice prefer is 30 to 33 degrees Celsius. And in fact, the basic studies where they put the mouse in a 20 to 23 or 30 to 33 in their open cages and ask them, where do you like to go? They will go to their thermoneutral temperature of 30 to 33 degrees Celsius. Another reason is what they have found. They have done studies in a mouse in the fields in their nests, and they've just placed the thermometers. And those nests are intentionally built to keep the temperature as high as they can, or as close to 30 to 33 degrees. That's why the mice actually build the nests and they stay huddled together. In fact, even in our mouse rooms, or most of the mouse rooms, that's the reason why you usually put 3 to 4 mice together to help them huddle together and increase that body temperature. So how does that impact on a mouse physiology? Well, that's where the things got really interesting. So the housing mice at thermoneutrality, which is here shown in red compared to blue, it significantly impacts mouse heart rate. So it almost halves it. So if you think in humans, if somebody would show up in a clinic with a half of their heart body rate, that would be a significant finding. It decreases the blood pressure significantly, decreases sympathetic nervous system activation, beta-2 adrenergic receptor signaling. And as an immunologist, I was really fascinated that the immune system in a mice housed at thermoneutrality is a little bit more augmented, meaning that there's less brakes on it, okay? And what was really cool is that in humans, if you actually have an infection, you will spike a fever. If you try to induce a febrile response in a mouse by giving an exogenous stimuli, either whether that being a ligand or a bacterial infection, it will actually drop in temperature. Okay, so it's unlike humans. However, if you place the mouse at thermoneutrality and you give them this challenge, they will spike the fever just like a human does. So those models together told us that there might be a relevance to modification of immune system here, and that we can use it for hopefully understanding human disease and metabolic disease. And so there is some assumptions that this is also relevant in humans. And so specifically the last point, where there's a very nice Dutch study, also although limited in numbers, but where they've taken individuals that have type 2 diabetes and placed them in a cold for just a couple hours a day, and then asked, is there a reversal of your type 2 diabetes? And in fact, the answer was yes. and that was correlated with a decreased inflammatory responsiveness of their immune cells when isolated from peripheral blood. So by using thermoneutrality, we think that at least we could potentially get us closer to improving a model and using this model to study metabolic diseases, including MASLD, in mice to a way that's more clinically relevant. So a few years ago, a graduate student in the lab was tasked with that. And so he did two key studies. He first asked, can you, by using thermoneutrality, induce atherosclerosis in mice? So here are the ApoE mice, which normally develop atherosclerosis. And so then if we put these mice at 30 degrees compared to 22 degrees, you can see that the atherosclerosis or plaques are significantly higher at 30 degrees. This correlated with increased inflammation in these plaques. But we were really not interested in ApoE mice because that's not the model we want to study. We want a black 6 mouse to develop atherosclerosis. And so Dan did these studies, placed C57 black 6 mice at thermoneutrality, fed them Western diet. And what you can appreciate is this is an atherosclerotic arch here, and that their plaques are significantly increased if these mice are fed Western diet and are at thermoneutrality. This also correlates with shift in your HDL, LDL, and VLDL, and increased inflammation in these plaques. So that was a step one. We like that we can push the model. Now, what happens with NAFLD or MASLD? Well, so Dan did these studies, and you can appreciate it if you place mice at 30 degrees and give them a high-fat diet, that there's this disease is significantly more robust. This disease occurs earlier. We can see that the NAFLD activity score is increased, that the hepatocellular damage is significantly amplified. And if we do RNA-seq on these livers, we can appreciate that there is a positive regulation of several pathways associated with MASLD progression. Excitingly, we can also start inducing fibrotic lesions in the liver just by feeding black 6 mice with a high-fat diet. So the next hurdle was, can you use this in female mice? And so the answer obviously is yes. So these are the female mice fed high-fat diet in regular housing. They will gain about 10 grams of body weight, but they do not develop metabolic disease. If you place these female mice at thermoneutrality, they will become severely obese and develop metabolic disease. This is, again, manifestation of their liver disease. High-fat diet fed female mice at 22 degrees don't develop much steatosis compared to the ones that are at 30 degrees, as well as hepatocellular damage. Now, what was really exciting for us is once we started teasing out what's driving this and isolating the immune cells, what we noticed is that at thermoneutrality, there's increased expression of IL-17. And then in fact, in both male and female mice, we have increased proportion of T cells that are infiltrating in the liver, and these T cells are producing more IL-17 and TNF concomitantly, suggesting that there might be more inflammatory. And so now we really asked, can we use this model to try to tease out the intricate pathways in these CD4 T cells to try to understand what regulates their cytokine production or pro-inflammatory pathogenic potential. So the first step we did is we placed the mice on a high-fat diet. We used IL-17 GFP reporter mice and sorted out the GFP positive cells and then did single-cell RNA-seq analysis. When we did these studies, we found that we can observe same things in both male and female mice, so we don't need to go into both comparisons. But if we look at this, what we see is that there's two flavors of Th17 cells in the liver, the ones that express this type of genes and the other ones that are missing those genes. And so we got excited to figure out what's the difference between them. In summary here, I'm just going to point out a couple of them. The exciting parts was the CXCR3 gene expression on it was increased in one subset of these T cells. And so that allows us to actually track these cells. The PKM2, which is key for cellular metabolism and glycolysis, and then interferon gamma, which is quite unusual to be found on Th17 cells or to be produced. So comparison of these cells immediately struck to us that they are what is known as pathogenic Th17 cells. But when we compare them to traditional MS models or EAE or Crohn's disease models of pathogenic Th17 cells, they had some similarities in their function, but they had about 300 genes that were very different and unique to the liver pathogenic Th17 cells. And so several of these pathways hinted us that this is downstream of the cellular metabolism. Well, we started digging into cellular metabolism. We were not the first ones to join this party. There's many papers now that are describing how cellular metabolism regulates immune function. And so what we wanted to do is to pick some of them that we thought we could uncover through our system and try to decipher them and see what they play. And so that's what I'm going to tell you for some of them. One, being on glycolysis, which actually positively regulate disinflammatory responses. The second one that I will tell you is on glutaminolysis, which we find that it actually negatively regulates disinflammatory pathogenic potential of Th17 cells. And the last one I'm not going to touch base upon today, but that's lipid metabolism, because we think that's also important in regulation of the pathogenic potential of these cells. So let's start with this. So when we first started looking at this, we saw that these cells, accrual of these inflammatory pathogenic cells, which are labeled here as Th17A, increases over the time of exposure to a high-fat diet, and that they're becoming more pathogenic, and that correlated positively with ALT levels. Now, what we also noticed is that these cells are cells that can actually co-produce multiple pro-inflammatory cytokines. So they don't just produce IL-17, they produce multiple pro-inflammatory cytokines at once. And we verified this in multiple models of MASL. So high-fat diet, NASH, Western diet, MCD diet, everything, because that's what the reviewers wanted us to do, so that we're not biasing the system. So how does this work? Well, the first question was, are these cells sufficient to drive the disease or to amplify the disease? And if you take conventional Th17 cells and you transfer them into an obese mouse, you can increase the disease pathogenesis. But if you take these highly pathogenic Th17 cells from the liver and you transfer them into a mouse, you can amplify that, suggesting that they do have the potential to drive this disease. And histologically, we can look at this. This is unusual that you get hepatocyte ballooning in this system, as well as macrophage infiltration is associated with that. So what drives the function of these cells? Well, RNA-seq would suggest that that is glycolysis. And in fact, we started verifying those things. And so you can appreciate there's all sorts of slew of glycolytic genes that are upregulated in these pathogenic Th17 cells compared to conventional Th17 cells. And what we did a really cool study is to say, well, are these cells uptaking glucose in vivo and in vitro more compared to conventional cells? And so we just injected mice with labeled glucose. And what you can see is if you isolate a minute later, you isolate T cells from the liver, you can see that the conventional Th17 cells take up glucose to this level versus the inflammatory ones take up a lot more glucose. So we think that this is the reason why they're driving more cytokine production. And if we do it in vitro cultures with these cells, we can recapitulate the same outcomes. So if glycolysis is important in this setting, obviously glycolysis should limit the inhibition of glycolysis should limit the MASL progression. And so that's what Maria did exactly. She first treated obese mice with 2DG to inhibit glycolysis, and we can reduce ALT, we can reduce infiltration or accrual of these pathogenic cells in the liver, as well as their ability to produce pro-inflammatory cytokines. But giving somebody inhibition of glycolysis in vivo, it's very hard. It's unlikely, it likely has multiple off-target effects. So she tasked herself to do a very challenging experiment. And that experiment is get the mice obese, isolate these pathogenic cells, treat them with inhibitor of glycolysis in vitro, or let them rest for a day, and then transfer them back in vivo to ask whether you can recapitulate these outcomes. And in fact, the answer is yes. So if you transfer the pathogenic inflammatory hepatic Th17 cells that are not treated, you have increased ALTs. But if you treat them with 2DG in vitro and then transfer, that is significantly suppressed. So that told us that these cells by themselves, via glycolysis, that this is an important pathway of driving MASL or NAFL progression. And so to as a proof of concept for this, we obtained the mice that are deficient in PKM2, and we could selectively deplete PKM2 in CD4 T cells. Unfortunately, we do not have a specific driver for just these pathogenic Th17 cells, so we cannot specifically target that. We can only target the whole T cell repertoire. And so if we do that, we actually can reduce the number of these cells in the liver, which is what's expected. We reduce their ability to produce pro-inflammatory cytokines if we delete PKM2 in them, and then we can reduce hepatocellular damage. So this finding was quite nice. We were pretty happy with it, and it added to the slew of other findings that glycolysis is an important regulator of immune cell inflammatory function. But we pushed this further. We started studying in human cohorts, and I'm just going to quickly go through this. We can discuss the human cohorts at the end, but these human cohorts have increased expression of Th17 polarizing cytokines and expression of chemokines that bind or signal CXCR3, which is important on these pathogenic cells. And if we do analysis of all sorts of things through the liver of the human subjects here, what you can appreciate is that if you have NAFLD, CXCR3 is increased, you have increased expression of TNF and interferon gamma, that there's increased infiltration of T cells, and specifically of these highly inflammatory pathogenic T cells in human cohort, and that these highly inflammatory pathogenic T cells, just like in mice, they have increased production of pro-inflammatory cytokines compared to conventional Th17 cells. We also find increase in PKM2, aldolase, and enolase, meaning the glycolytic pathway in these individuals with disease. And this also nicely correlates with the progression of a disease on a clinical side. So we were pretty happy with this. We think this has some relevance, but we still really don't know glycolysis is essential for cell survival. It's essential for many, many different pathways. And we're really interested to figure out how these cytokines are being produced and regulated. And so now I'm going to switch to the other topic here, which we covered glycolysis, but this one we'll cover on glutaminolysis. And so we think that we focused on glutaminolysis as one of the amino acid using pathways or metabolism, because we saw some effect that it actually could be doing the opposite. So finding this balance might be important in these T cells. And so why glutaminolysis? Well, glutaminolysis, if you look at it, this is the glycolytic pathway, this is your mitochondria, and glutaminolysis pathway is down here. It makes intuitive sense to study additional pathways in T cells. Well, T cells also utilize different metabolic pathways for a variety of functions, so not only glycolysis, several of them listed here, including glutamine. Now, glutamine metabolism via GLS1, which is down here, is implicated in establishment of specific CD4 T cell subsets, including Th1 and Th17 cells, and glutaminolysis is upregulated during T cell activation. We think that these T cells are activated in progression of NAFLD or MASLD, and so understanding how glutaminolysis deals with this is very important. Now, we also know from one very basic study that glutamine supplementation reduces pro-inflammatory marker expression in the liver and improves insulin sensitivity during obesogenic stress. So this gave us enough rationale to try to figure this axis out. When we went back to our human cohort and asked, well, again, what we see that in addition to inflammation and IL-17 signaling pathway, we see that glutamine family of amino acid metabolism and regulation of glutamine transport is also upregulated in the individuals with NASH. And we see that the two key processes, GLS1 and OGT, are also upregulated in this setting. So what happens in mice? Well, if you place a mouse on a high-fat diet and you ask how much glutamine is available in that animal in the liver, actually, it's decreased, and those levels keep decreasing throughout the progression of a disease. So this is 12 weeks on a high-fat diet. This is 27 weeks on a high-fat diet. You start seeing similar effects on the MCD diet, which is another model of fatty liver damage. And in fact, if you take the mouse on a high-fat diet and you ask glutamine levels and ALT, they are negatively correlated. So more glutamine you have, less ALT you have, etc. So with that in mind, with the first study that Kay did, a student in lab. So what he decided to do is to actually ask, can we, by giving exogenous glutamine, reverse the hepatocellular damage in obese mouse? And so here he places the mice on a high-fat diet for 20 weeks, and then we just treat these mice for 2 weeks with glutamine. So this is not pre-treatment. We're not skewing anything. It's just at the end of the treatment. And so what he can see is that we can, by giving exogenous glutamine now in the liver, we can recover or increase some of the glutamine levels. If you notice, this is to about 25. Normal glutamine levels are about 30 to 40, so we do not fully recover what you would have in a chow diet, but we can push up the limits of glutamine in there. And I don't know if we keep treating him longer or with a higher doses of glutamine, maybe we would get there, but we have to be careful because too much glutamine is also not safe. So what this treatment does, it doesn't impact your body weight. So that's good. So we are not comparing mice that are of different body weight. And then if we ask what happens with hepatocellular damage, in fact, if you give mice these glutamine, most of the fat accumulation and inflammation in the liver goes away. So what you can see here is that the liver weight is reduced, triglycerides in the liver are reduced, and the hepatocellular damage is significantly reduced. So just by giving glutamine, we can actually help or restrict this progression of MAS. So does glutamine do anything to CD4 T cells? Well, I gave you the whole spiel before that it does, but we wanted to see it in this system. So he went into the liver of these mice and he can see that these CD4 T cells in the liver of the mice that are treated with glutamine produce less TNF, interferon gamma, and IL-17. But this is not seen in CD8 T cells. So this seems to be a pattern specific to CD4 T cells, okay? If we take these mice and then give them exogenous bolus of anti-CD3, which will activate your T cells and induce T cells to produce cytokines, if you do that in vivo, the ones that are treated with glutamine produce a lot less pro-inflammatory cytokines. So this not only is a liver unique, but it also impacts your systemic production of pro-inflammatory cytokines. And then lastly, he asked in vitro if you can manipulate the glutamine levels. Do you impact the cytokine production? And you can see that as you increase glutamine levels in vitro in T cell co-cultures, that you actually reduce IL-17 production by these T cells. So this was a nice finding. So now we have two sort of findings that we're hoping to connect. Glutamine helps you with your mast cell pathogenesis, but then glutamine also restricts Th17 cell IL-17 production. So are they linked and can they be linked? And how are they linked? So again, this is just a schematic for glutamine. We pull a bunch of cells from the liver and ask which cells express the most GLS1. And what we see is that really the Th17 cells are the highest producers or expressors of GLS1, which is a key enzyme that you need to break down this glutamine. And if we take the GLS1 fluxed mice, we put them on a CD4 CRE. If we stimulate these mice with anti-CD3 to see now if you do not have GLS1 in T cells, can you induce pro-inflammatory cytokines? In fact, what we see is that activation of T cells in general, in the absence of GLS1, amplifies cytokine production. So you need GLS1 to restrict this pro-inflammatory milieu. And so we went on, did the same studies as before, pulled the T cells out from GLS1 sufficient and deficient mice in CD4 T cells, ran RNA-seq, and what we see, gene expression fits our profile, that there is a difference in Th17 cell differentiation and interferon gamma production. And so then Kay said, well, what happens to these mice if you put them on a high-fat diet? And you can appreciate here that these mice, sufficient or deficient, are fed high-fat diet. There is no difference in a body weight, so they're equal. There is some difference in, no difference in liver weight that you see, but there is a difference in ALT. So T cells, GLS1 in T cells is not sufficient really to regulate your hepatic steatosis. That is likely happening through impact of glutamine on hepatocytes, which it has a significant impact, but the inflammation itself that is happening in the liver, it does seem to be occurring through CD4 T cells. So to test that, he went further, pulled the CD4 T cells from these mice, again, from the livers of these mice, and he can see that if you give if you use GLS1 deficient mice with in a T cells, that you have increased TNF, increased interferon gamma, and increased IL-17A, but you do not have the same effects in CD8 T cells. So again, showing some specificity towards Th17 cells. And lastly, he wanted to ask, if you give this exogenous glutamine to a mouse, is this dependent on GLS1 in T cells as far as the hepatocellular damage? And so he set up the study here where he would treat GLS1 sufficient and deficient mice with glutamine, exogenous glutamine, and these are the 3 bars here. The key points here is that if the mouse just receives glutamine, you drop in this triglyceride accumulation, the ALT drops, but if you do not have GLS1 in your T cells, the ALT does not go down, suggesting that the GLS glutamine has 2 benefits. One is removing or reducing the hepatocyte steatosis through likely function on hepatocytes, but it's important in reducing your hepatocellular damage by modulating the CD4 T cell cytokine production. So these are the 2 pathways that I wanted to touch upon, but this is a cellular metabolism of a cell, right? And so we're scratching the surface of all of this, like very, very slowly, okay? There's many targets that we want to go. We're interested in, obviously, there's a lots of therapeutic targets out there for targeting cellular metabolism. So we're exploring these, hopefully to give us a more clinical relevance in how to approach this, because it's going to be very difficult to target a specific cell and cellular metabolism in a specific cell. So we need to know more and more mechanistically of what it's doing. And, if we target only one pathway, it's likely that that cell will adapt and activate the alternate pathway that you do it. So most likely the outcome is that we will have to use multiple targeting therapies. So in this instance, maybe inhibiting glycolysis at the low level, but actually amplifying glutaminolysis in order to reduce this CD4 T cell cytokine production. So in summary for this part, before we switch to something else, is that what I was hopefully able to convince you is that the obesity promotes this hepatic accumulation of a distinct Th17 cell subsets, okay? They don't all come in the same flavor. We see this in humans as well. What we term them inflammatory hepatic Th17 cells because we think they are the most robust pathogenic producers of these cytokines. That the CXCR3 axis, which is the chemokines that recruit these cells to the liver and keep them there. I didn't show you the studies today, but if we actually take the mice from that are obese, bring them back to a chow diet so that they lose all this weight, these cells leave the liver. So this axis is important in keeping these cells in the liver, and that's what it allows them to produce these cytokines in that milieu that then impacts presumably hepatocytes. I also didn't show you that if we co-culture these cells with the hepatocytes, we can induce a lot more in vitro, a lot more ALT release from these hepatocytes compared to conventional Th17 cells. And then that the glutamine actually, or axis in these cells, works in the opposite way, that actually it restricts your inflammatory cytokine production, that glutamine has beneficial therapeutic effects, not only on T cells, but we think it also impacts hepatocyte function as well. Unfortunately, we're not very good with studying hepatocytes. That is, we're immunologists, so we're sort of putting that aside. But I think there is a real utility in that part to be studied. And then that we also see some similar effects in human mast cell progression. So what we think is really that the cellular metabolism of these cells being skewed, it's important in regulating the functional outcome in a disease-specific manner. So for the last couple of minutes, I just wanted to jump onto something else. And the premise of this really comes down to that this is all nice that we can target the disease later on in life. But what can we do to learn what happens early? Because earlier we find out when these changes are occurring, the targeting is better. It's likely that we will limit the disease progression and establishment. Once the disease is established, it's very difficult to treat. So here, we started, a graduate student in the lab started working on something that we're trying to figure out how that these pathogenic traits are transmitted from a mother to an offspring. And if you go through some old literature, you can see that actually there's a U-shaped curve, at least for type 2 diabetes preponderance, that the weight gain by itself, so if you're on a very low side of the weight, that you have a higher risk for type 2 diabetes, and that goes the same way for a severely obese. And in fact, if you're in the middle there, you're pretty protected, okay? And that probably, if you go clinically to lots of things, you will find a similar spectrum for a variety of diseases. Infectious diseases are one of them, that if you are normal weight or maybe even slightly overweight, you're likely to handle this insult better compared to if you're undernourished or overnourished. So what does this mean with obesity? Well, there's some premise out there that children of parents with obesity are more likely to develop obesity themselves. These studies are very challenging because obviously in those households, the food consumption and intake is not healthy, so it's likely that these kids will get it from that. But they have done some studies where actually one child in a household eats what is presumably a regular food, and it still doesn't slow them from becoming obese, suggesting that there could likely epigenetic modifications at play here. Rodent models, studying of them, have shown increased predisposition to liver and kidney disease if the mother is obese. And so immunologically, we're quite interested because if your mother is obese, the offspring is more susceptible to development of autoimmune diseases. There is a reduced survival when infected with gram-negative bacteria, for example. We know that there's a dysregulated response to toll-like receptor stimulations. And what we are excited in is the alteration of CD4 T cell populations. So for all of this, I'm not going to give you a mechanism. I'm just going to show you what we have found. So Jen, MD PhD student in the lab, she was tasked with one of these to actually set up the model. And so here we're blessed with being able to use the thermoneutral housing. So we put the female mice at thermoneutrality and either feed them chow diet or a high-fat diet. And once they become obese, then we mate them with the lean males, okay? And we intentionally use lean males because we only want to study the maternal effects. And so what happens out there, then we ask once they're pregnant, what are their outcomes to see whether our model would actually resemble what you see in humans. And in fact, if we look at the litter size is pretty comparable. Weight at birth, it's a little bit lower in HO stands for obese mothers. And so what we look from, they're low for gestational weight, which is what you can see also in the clinic. But the survival, this was the most surprising part, is that the survival of these pups that came from an obese mother was significantly lower. So this tells us that this impacts something on their early survival, and this all happens within the first 4 days. Once they make it through those first 4 days, those pups are normal. And so this makes these studies very challenging because one, obese mice are very difficult to get pregnant, just like obese humans. So there are issues with pregnancy and reproduction, but then the ones that do get pregnant, you do not get a lot of pups. So Jen was asked what the next step is. If you let these mice go on a switch them offspring from obese mother and you switch them to a regular diet, they do not develop metabolic disease. Their liver looks normal. In fact, all they have is increased propensity to accumulate a little bit more white adipose tissue. So I'm not showing you that data, but it's in that paper that's published. But where the really cool parts are is that these mice are a lot more sensitive if you challenge them with a high-fat diet. And by a lot more sensitive, that's what Jen did, is she took these chow-fed lean or high-fat diet of these mothers and then placed them, fed the offspring essentially with either chow diet, high-fat diet, or high-fat diet, okay? And what you can appreciate is that the ones that came from an obese mother and were fed high-fat diet, they significantly gained more weight compared to the ones that came from a lean mother but were fed high-fat diet. That this correlated with a percent of total body fat, and that they have increased in glucose tolerance or glucose metabolism, if you want, that their liver weight was increased, and that, again, we have a very low numbers, but that their hepatocellular damage was significantly worse or exacerbated in this setting. And so we wanted to know, when do these changes occur? Because again, this is what we are seeing once these mice are adults because of us feeding them a high-fat diet. But do you see some of these inflammatory changes early on? And so Jen set up this study where we actually went in and looked for serum endotoxin levels in an obese mother, and as expected, there is increased systemic endotoxemia or metabolic endotoxemia. This also correlates with increased IL-6 levels. But then we went further and we asked what happens in the amniotic fluid. And we wanted to do this because we think really this gestational period in humans of 9 months or mice of 3 weeks, it's probably the key area where you have a continuous signaling that may change these epigenetic modifications or cues. And in fact, what we see is that there's increased endotoxemia in the amniotic fluid of these obese mothers, and that if you take the MEFs or cells from these pups that are born to obese mothers and just stimulate them with TLR ligands, they produce significantly more IL-6 compared to the ones that are coming from a lean mother, okay? So suggesting that even at day 10 of gestation of a mouse, which is very early, these cells are already ready to be more pro-inflammatory in this setting. So they're skewed how they respond compared to a lean setting. So we went further and we took this amniotic fluid and we just asked for proteomic analysis. What do we see? And if you can appreciate here, so one is this is a control, this is obese mother, and the pathways here that we see from a proteomics, what I'm showing you is that there's lots of it is inflammation associated, okay? So this amniotic fluid is highly pro-inflammatory if a mother is obese. And in fact, you can pull different pathways that you can study, and that's what we are trying to do now, to try to narrow down which of these could be done and how we can manipulate and target them potentially in utero and ask what their outcomes would be. Now, this is also relevant to humans because we have set up a human cohort as well to try to do similar studies. We obviously cannot target the amniotic fluid unless there are issues with pregnancy and collection. But what we have seen in pregnant mothers, these are the lean ones and these are the obese ones, that the obese mothers have increased systemic levels of pro-inflammatory cytokines, that subcutaneous fat thickness is not different in these offsprings. But what we do see, if your mother is obese, that these babies by 9 months, their increase in body weight is significantly upregulated compared to if the mother was lean. Actually, that slows down, suggesting that unfortunately for keeping these babies, and we're now studying the liver disease in them as well, to perform these studies, it's challenging because they do not, as I was telling Jackie, they do not stay still after 12 months. So we cannot really do the nice analysis in the liver and fat deposition in the liver, but this is the direction we're going in. So we see some of the impact on metabolic disease if a mother is obese as early as 9-month period. And so in the summary for this part, because it is time to end this anyway, is that the thermoneutrality allows us for induction of severe obesity in female mice, that maternal obesity correlates with dysregulated in utero inflammation and skews immune responsiveness in the offspring. This presumably is linked to the amplified metabolic disease severity. Now, what we are really interested in is how this is happening and can we modify this? And in mice, we can, because we can do embryo transfers. We can also do amniotic fluid transfers. And that's exactly what we're trying to do now to see whether the outcomes are modified if we jump early on. It is still far-fetched, but there are strong push, especially for the women that are undergoing preterm birth, where they're building outside a womb, essentially. That's genetic, that is engineered specifically to allow the babies to survive longer. That's artificial womb with specific modulations, etc., that will come in for a preterm birth, hopefully in the near future for those babies. So can this similar manipulations be applied towards a metabolic disease or whatnot? I don't know, but we can certainly try to see how it goes. So with that, I'll just end it. Thank all of our collaborators, my great lab members. There's a slew of collaborators without whom we would not be able to do this, and our funding sources, and I'll take any questions. Thank you.
Thank you.